Hello, you're welcome to Medical Sciences Made Easy by Naphtali Muhumuza, and today we want to talk about translation, which is part of protein synthesis. That's why we are saying translation or protein biosynthesis. And I always remind you to subscribe if you haven't subscribed. Like the video and always share your comments. So translation, which is part of the central dogma. In the beginning, we saw replication, we saw transcription, and today we want to see how we use the products of transcription, that is mRNA. That whenever you have messenger RNA, transfer RNA, and ribosomal RNA, that how do we utilize them by using information that is the genetic code on mRNA. The genetic code, which is in the form of codons, how do we translate it, or how do we use it to form a new protein? And this one, it occurs in the presence of ribosome. And these ribosomes are found in the cytosol. So this process occurs in the cytosol, where we find our ribosomes being as machinery. That whenever you have the genetic code or the codons in the form of codons, we want to translate it or we want to transfer it into a protein in the presence of ribosome in the cytosol. And that is what we call translation. Translation is the interpretation of the information in the form of genetic code on mRNA in the presence of ribosomes, and it occurs in the nucleus of the cell. No, it occurs in the cytosol of the cell. And this translation, which is using ribosomes as machinery, that ribosomes, we know they are made up of larger subunit and smaller subunit, whereby in prokaryotes, this one forms a 70S, and when it is dissociated, it forms the 50S ribosomal subunit and 30S ribosomal subunit. This is in prokaryotes. Then in eukaryotes, those are multicellular organisms. We have the larger ribosomal subunit and the smaller ribosomal subunit, and all of them form 80S, whereby this is the 60S and this is the 40S. So these are the different structures of ribosomes, both in prokaryotes and eukaryotes. And these, they always have what we call the P site and the S site. This is the P site, that is peptidyl site, and the S site, which is amino acid site. Also, this one has the P site and the S site. So this is S site, this is P site. So this is the ribosomes that we're going to discuss in this session, acting as machinery for the synthesis of new proteins. And what are the requirements of translation? Among the requirements, we shall need transfer RNAs, we shall need mRNAs, which are carrying codons, and the transfer RNA carries the anticodons. And these anticodons are the ones that specify for the amino acid to be added. Then another requirement we shall need is enzymes, and these enzymes include initiation factors, include elongation factors, include termination factors. And even so, many enzymes are required. Then we shall also need, the way we said, we need ribosomes. So these are the requirements, and also we need energy in form of ATP and GTP. So these are the requirements of translation, that whenever translation wants to occur, we need factors like enzymes, we need ribosome in the cytosol, we need ATP and GTP as a source of energy, and we need transfer RNAs and messenger RNAs, whereby we have said the transfer carries the anticodons, whereas the mRNA carries the codons, and it is the codon that specifies the amino acid to be added by the transfer RNA. And the stages of translation, it occurs under different steps or stages, whereby the steps we have amino acid activation, we have initiation step, we have elongation step, and termination. So there are 4 stages or steps that occur during translation. That is, the first one being amino acid activation or charging. We have initiation. Number 3 is elongation. Number 4, and after we shall also talk about what we call post-transcription modification. So we are going to discuss these steps using these requirements listed here. And the first step which we want to talk about is amino acid activation, whereby when we have a transfer RNA, this one being the 5' end and this one being a 3' end, we are going to see that the 3' end of the transfer RNA ends with cytosine adenine, which is coupled with hydroxyl group. And this transfer RNA, that it is activated or by charging, or we add what we call an amino acid. And whenever we add an amino acid, this requires energy. This addition of amino acid at 3' end requires energy, and this energy is supplied by ATP. But whenever I have adenosine triphosphate, is split into adenosine monophosphate plus a pyrophosphate. And this one occurs in presence of a cofactor known as magnesium ion as a cofactor, whereby we are adding amino acid specified by the codon messenger to form amino acyl transfer RNA complex. And this one, to form this, we're going to see addition of this amino acid at 3' end of the transfer RNA by removing of the hydroxyl group, forming amino acyl transfer RNA complex. This is the 5' end, then it is added at the 3' end, ending with cytosine adenine. So, and this process of amino activation occurs in the presence of enzyme known as amino acyl transfer RNA synthase. As we know that synthases are named according to the product they are catalyzing, and because we are forming amino acyl transfer RNA, we are seeing the enzyme is amino acyl transfer RNA synthase enzyme, catalyzing, whereby it needs energy in form of ATP. So this is what we call amino acid activation or amino acid charging, because before we begin the process, we needed to add an amino acid on the transfer RNA. And after the first step of amino acid activation, we begin with the second step, which is initiation. And initiation step, it begins with dissociation of the ribosome, whereby if it is in eukaryotes, we see the 80S dissociating to give 60S and 40S. If it is in the prokaryotes, we see 70S dissociating to give 50S and 30S ribosomal subunit. So this is the first step under initiation. That under initiation, we need to separate the larger subunit and the smaller subunit. That is what we are calling ribosome dissociation in the cytosol, that whenever in the eukaryotes we are having the 80S, it is dissociated 60S and 40S. If it is prokaryotes, which are smaller microorganisms, for them it is split to 50S and 30S ribosomal subunit. So that's the first step under initiation. After dissociating the ribosome, this is what we are saying, that in the case is eukaryotes. If this is in eukaryotes, we are seeing this, which is the 80S being split. This is the 60S having the P site, it has the P site. This is the S site, this is the P site. This one is, when it is in eukaryotes, it forms the 40S and the, so this is the 60S, this is the 40S ribosomal subunit. And after forming these ones, all of them have the P site and the S site. So this one has the P site and the S site. After this, the process which we are calling dissociation, the initiation factors that catalyze the dissociation, we have elongation factor, eukaryotic initiation factor 1A, and factor 3. So the ones or the factors that initiate this process of dissociation is eukaryotic initiation factor 1A and factor 3. Initiate doing that. Whereby in prokaryotes we have initiation factor 1 and factor 3 doing the work of the. So after dissociating it, then we form the 40S ribosomal subunit, and this 40S is going to help us to form what we call pre-initiation complex. Pre-initiation complex, whereby we are going to add the mRNA. But before we add the mRNA, how do we form the pre-initiation complex? Is that after charging the amino acid, the transfer RNA, and we know the first transfer RNA that is formed in eukaryotes, it is MET, which stands for methionine transfer RNA, which is always the first amino acid in all proteins. So if this complex is formed, we are seeing it is going to be formed in the presence of ATP, and it comes and binds in the P site. This is why we called it the P site, whereby it comes. So we're going to see the MET transfer RNA coming into the P site of the smaller ribosomal subunit, and this one occurs in the presence of initiation factor, that is eukaryotic initiation factor 2, in the presence of GTP. Even these ones occur in the presence of GTP, which is providing energy. And this eukaryotic initiation factor 2 in presence of GTP is the one that forms collectively. Collectively, these guys is what we call 43 initiation complex, 43S initiation complex. So we have converted the pre-initiation complex, and we have formed what we call the 43 pre-initiation complex, whereby the 43 pre-initiation complex is being formed by eukaryotic initiation factor 2 in the presence of GTP, whereby it is adding the MET transfer RNA in the P site, forming the 43 pre-initiation complex. And it is this 43 pre-initiation complex that is used to form the 48S initiation complex. And how do we form the 48 initiation complex? Is we have already in the P site, we already have our MET transfer RNA that is in eukaryotes, and then it was without messenger RNA. But at this point, to form the 48S, we see the binding of messenger RNA to the 43 pre-initiation complex. So the messenger RNA comes and binds to the 43 pre-initiation complex. And how does it bind? It needs, in prokaryotes, we know that they have what we call the 5' prime cap. And how it happens in this prime cap, we have what we call a protein known as cap binding protein. Cap binding protein, or you can call eukaryotic initiation factor 4, which recognizes the nucleotide sequence in the cap site, which is a pre-initiation factor for recognition of AUG. And we see here there is AUG, which is the first codon that codes for methionine amino acid. So when the messenger RNA binds to the 43 pre-initiation complex in the presence of a protein known as cap binding protein, or in eukaryotic initiation factor 4, that's when we form a 48S initiation complex. And it is this 48S initiation complex that is going to bind with the cleaved off 60S. So we are going to see these two which have been formed. This one which had been cleaved off is going to combine. So this one combines together with the 40. This is the 48. This is the 60S ribosomal subunit. This is the 48 the initiation complex. And these ones, when they combine together, they form the 80S initiation complex. And our major aim under initiation was to form the 80S initiation complex, whereby you combine the 60S ribosomal subunit that was deassociated together with the 48S initiation complex that is formed after adding the messenger RNA. And that is in the eukaryotes. In the prokaryotes, we are going to see that for it doesn't have the 5' end cap. And the methionine, which is added in prokaryotes, instead of methionine, we are saying in the prokaryotes, for it, the one which is added on transfer RNA is N-formylmate, N-formylmate transfer RNA. That is in eukaryote, in prokaryotes, and for it, it does not, we see the pre-initiation nucleotide being the cap binding protein, which we are calling eukaryotic initiation factor 4. That is in eukaryotes. But in the prokaryotes, we do not have this, the 5' end cap. So what happens, the pre-initiation nucleotide for the transfer RNA to detect the start codon is what we call the Shine-Darugano sequence. Shine-Darugano sequence. It is a nucleotide of bases that enables the transfer RNA in the P site to recognize the start codon, which is AUG. So in eukaryotes, it is a cap binding protein, but in prokaryotes, it is the Shine-Darugano sequence, which appears before AUG. So we find in eukaryotic, the cap binding protein is in here, but in prokaryotes, it is Shine-Darugano sequence that enables the transfer RNA carrying methionine N-formylmate to recognize the start codon, which is AUG. So this is what we call initiation step. So finally is that when this one, 60S ribosomal subunit, combines with methionine transfer RNA complex after binding of the messenger RNA to form the 48S initiation complex, this is what we have formed, which we are calling 80S initiation complex. And that is the one that marks the end of initiation step. So we have finished amino acid activation. We have also seen elongation. And what factor that enables association of this with this to form 80S complex is what we are calling elongation factor 5. Eukaryotic initiation factor 5. Eukaryotic, E stands for eukaryotic. So eukaryotic initiation factor 5, it combines the 60S ribosomal subunit which was cleaved away together with the 48 initiation complex to form 80S initiation complex, and that one that characterizes this reaction is eukaryotic initiation factor 5. And this eukaryotic, with this one to be formed, we need eukaryotic initiation factor 5, which is always bound to GTP to provide energy. And this one, it cleaves away the eukaryotic initiation factor 1, factor 2, and factor 4 back into the cytosol to go and initiate other processes. So this is what ends with the initiation. So from here we are going to begin elongation steps. Under initiation, the first step is the dissociation of the ribosome, and after dissociating, we see the hemoglobin transferrin A binding to the P site in the presence of elongation factor 1 and factor 3 to form 43 pre-initiation complex. The third step under initiation is the formation of 48 S pre-initiation complex, which in eukaryotics we need cap binding protein, in prokaryotes we need shiny organo sequence to enable the messenger RNA to bind to the smaller ribosome, and then for the mate transfer RNA to recognize the start codon, which is AUG. And after forming the 48 subunit initiation complex, now we bind the 60th which went away together with the 48 to form back the 80S initiation complex in presence of eukaryotic initiation factor number 5, and GTP providing energy, and then other initiation factors are recycled to go back and activate others. So that is what we can talk about under initiation. Then the third step is elongation. The third step we have is elongation. And this elongation, as the way it suggests, is we are now going to prolong the chain. So we already have here, this is AUG, and this AUG, we bring in the charged transfer RNA with an amino acid, and this one is mate, and is going, since this is A, is going to bind with anticodon uracil. Then uracil here binds together with adenine, then guanine binds together with cytosine. So when they come, we have formed this is the first amino acid in the P site, then this is the A site. So during elongation, it occurs in 3 steps. One, it is the addition of the next incoming amino acyl transfer RNA carrying the amino acid specified by the codon on the mRNA. Then the second step, we are going to see the formation of peptide bond. Then the third one, we are going to see translocation. So these are the major 3 steps that occur under elongation. And the first one is the next incoming amino acyl transfer RNA binding to the A site. We have seen during initiation, the first amino acid, which is methionine, is added into the P site. It is in the P site, what we call peptidyl site. But during elongation, the second incoming amino acid specified by the codon on the messenger RNA, binds to the amino acid site. And when it comes together with its amino acid, and then here there is another codon, it comes with a second amino acid, then it binds by its anticodons, and these codon together with anticodon, they must be complementary, and they are the one that specifies the second amino acid canon. So we are saying this is the first amino acid, this is the second, and after coming and binding into the S site, what happens? We need an enzyme known as peptidyltransferase. So peptidyltransferase enzyme comes and forms the peptide bond between the first amino acid with the second amino acid. So it facilitates the formation of the peptide bond. And this peptide bond formed between the first and the second amino acid in the P site and the S site respectively is catalyzed by an enzyme known as peptidyltransferase. Peptidyltransferase enzyme. And this peptidyltransferase enzyme requires eukaryotic elongation factor 1 alpha together with GTP to provide energy. So we are saying formation of peptide bond between the amino acid first in the P site and the second amino acid in the S site, catalyzed by peptidyltransferase in the presence of eukaryotic elongation factor 1 alpha together with GTP, which provides the energy, leading to the formation of the peptide bond. And after forming the first peptide bond, we cleave away the first transfer RNA carrying methionine and is taken back to the cytosol to bring more to be charged. And then when this is cleaved away, we form the end result of this. After cleaving away the first transfer RNA, we form dipeptide transfer RNA in the A site. So this one is in the A site because we cleave away this transfer RNA. And after forming that first peptide bond in the presence of peptidyl and under inhibitors, we are going to see which drugs target, like lincosamides and chloramphenicol, inhibiting peptidyl transferases, hence stopping the formation of the peptide bond. Then first, the last step is translocation and elongation. And translocation, trans means movement. So we're going to see the mRNA remains stationary, whereas the ribosome moves one codon ahead or jumps one codon ahead, and then the mRNA remains stationary. So we are going to see at this step is elongation, whereby during elongation step, we have our 5' end, this is the 3' end. The AUG, which was in the P site, comes outside. This is AUG. Then another codon, which was in the S site, comes to the P site. This is the P site, this is the S site. So this one comes in the P site, and there is another codon, other more codon is coming in. So this is what we call translocation. This is translocation, whereby translocation is movement of ribosome one codon forward to create space of a site, because we need more incoming amino acids to bind in the amino acid site. So that's why the ribosome moves one step ahead to create or to empty the space of a site to allow more incoming amino acids to bind. So when it translocates, in the presence of an enzyme known as translocase enzyme, in the presence of course energy provided by GTP, it moves one step ahead and enables the dipeptide transfer RNA to move into the P site. So we are going to see our dipeptide transfer RNA moving into the P site, emptying, creating the space in the A site, what we call the amino acyl site. And when it is created, it allows the third incoming amino acyl site, amino acyl transfer RNA to come and bring the third amino acid. And we're going to see it is going to come and bind inside here. And when it binds, it comes with another amino acid specified by the codon on the mRNA. And when they are complementary, it brings the third. And when the third reaches in the S site, remember there is peptidyltransferase and elongation factor alpha, 1 alpha. It is going still to create another formation of the second peptide bond between the second amino acid and the third amino acid. And so it leads to formation of a tripeptide. And after forming this peptide bond here, it cleaves away this transfer RNA and exits and goes back at ribosome. And after exiting, we form our tripeptide here. And after forming this tripeptide in presence of peptidyltransferase and GTP, then translocase moves another in presence of GTP, which is providing energy. The ribosome again moves one step ahead to another anticodon. So we are going to see AUG continues to move away as we are translocating the ribosome. So we are seeing another codon here. Then the fourth codon comes in the P site, and the S site becomes again empty to allow another fourth incoming amino acid. So we are going to see in the P site now, this one moves in the P site because we have moved one step ahead. We are going to have now our transfer RNA carrying 3 amino acids. Then we bring another one. And when another one is coming, so this step of elongation continues repeatedly by action of peptidyltransferase and translocase enzyme in presence of ATP and elongation factor 1 alpha. All of those are the ones that are characterizing this elongation step, and it continues repeatedly until it continues translocating and reaches a stop codon. And this is what we are going to discuss under termination step. So we are saying the last step, as we are going to conclude, is that the last step is termination. Termination. And termination, we are saying it occurs when we form a long polypeptide chain. We continue repeatedly by the action of translocase and peptidyltransferase. We form a long chain of amino acids. And when another one which is incoming, it finds there is a stop codon, or what we call a nonsense codon, which we normally say, you go away, you go away, you are away, you are gone. So these are the stop codons, UGA, UAA, and UAG. There are 3 stop codons. So whenever we are elongating, and then the S site, or the codons in the S site, we find it has formed a termination signal, which is a nonsense codon, then the release factor comes into play. So we are going to see that during termination, done by the 3 stop codons, or nonsense codons, which is UGA, UAA, and UAG. And when they come into play, the presence of these stop codons in the S site, so this is A, that one can be UAG, UGA, which is a stop codon. Whenever it reaches in the S site, it is detected by the termination signal, and this termination signal leads to release of release factor. Release factor, whereby in prokaryotes or prokaryotes, we have release factor 1, factor 2, and factor 3, Then in eukaryotes, we have a single release factor. And these release factors, they come and work together to neutralize the action of peptidyltransferase. Because whenever peptidyltransferase is present, it's going to continue to make more peptide bonds. But what happens, the release factor comes into play, and we know that stop codons do not code for any amino acid. So the transfer RNA comes empty because there is no amino acid that is specified for these three nonsense codons. And because there is no amino acid that is specified by them, we're going to see the release factor coming into play, hydrolyzing peptidyltransferase. And when peptidyltransferase is hydrolyzed, no more formation of the peptide bond, and it will lead to the release of the polypeptide chain. So the polypeptide chain is released by the release factor outside the P site, and it is the one that is going to undergo post-translation modification. And this is the one that will mark the end of translation. So we have seen translation occurs in 4 steps. Don't forget them. Amino acid activation, initiation, elongation, and termination. And we have seen all the processes involved in there, whereby in amino activation, we have seen enzyme amino acyl transfer synthase, transferase synthase, catalyzing the addition of amino group at a 3' end ending with CCA base nucleotide, forming amino acyl transfer RNA, whereby the first one is always mate transfer RNA in eukaryotes and N4 mate in prokaryotes. Then we have seen the second step was initiation, which was divided into dissociation of the ribosome, followed by formation of 43S pre-initiation complex, followed by the binding of messenger RNA to form 48 pre-initiation complex, which binds with the 60S to form 80S initiation complex, that is in eukaryotes, and we have seen even the factors involved. And with that one was marking the end of initiation, whereby we said the initiation sequence in eukaryotes, we have the CAP binding protein, or what we call eukaryotic initiation factor 4. Then in the prokaryotes, we have Shine-Darugano sequence, which is a base of nucleotide full of purines that enable the transfer RNA to recognize the start codon, which is AUG. Then we have ended up with elongation, whereby we have seen elongation occurs, the incoming amino acyl transfer binding in the A site, then formation of the peptide bond by peptidyltransferase enzyme, and translocation catalyzed by translocase enzyme, both in the presence of GTP to provide energy, and also presence of eukaryotic elongation factor 1 alpha, which was enabling the formation of the peptide bonds, resulting into formation of a polypeptide, which can be folded to form proteins. And finally, we said, as it continues to translocate one step or one codon ahead, when it reaches the stop codon, what we call the nonsense codon, UGA, UAA, and UAG, we are able to provide the release factor, which comes into play. And the release factor in prokaryotes is 1, 2, and 3. Then in eukaryotes, it is eukaryotic release factor, which hydrolyzes peptidyltransferase and causes the association of the last transfer RNA to release the polypeptide chain. And this is what we can talk about under translation. Finally, is to look at the inhibitors. Inhibitors of translation. And under translation, there are different inhibitors that play, or the drugs. We are looking at inhibitors of translation, and one of them is aminoglycosides, aminoglycosides. We shall have drugs like tetracyclines. We shall have drugs like streptomycin. And all these drugs bind to smaller subunit, that is 30S of the prokaryotes. And when they bind to 30S, we're going to see the aminoglycosides like gentamicin and streptomycin preventing the formation of the pre-initiation complex. These guys inhibit formation of pre-initiation complex. And without pre-initiation complex, we cannot synthesize proteins. And this one, the tetracyclines, for them, they inhibit the binding of MET, N-formyl MET transfer RNA on the ribosome, on the messenger RNA. So they bind to the 30S ribosome subunit, preventing the initiation step, whereby some bind to the stops the formation of this complex, and others prevent the binding of the MET, N-formyl MET, into the P site, and thus inhibiting the translation. So these are drugs. Another category, we are going to see drugs like macrolides, and a good example is erythromycin. And this erythromycin, we have azithromycin. These ones bind to the 50S, which is the largest subunit, and prevent translocation. Or they inhibit. So they do not allow the movement of ribosomes one step ahead. And when you do not allow translocation, elongation step is terminated. So these guys, macrolides like erythromycin, azithromycin, inhibit elongation step by inhibiting translocase enzyme responsible for the movement of ribosome one codon ahead. Then another drugs we can look is the riconcamides, like clindamycin, and another drug known as chloramphenicol. And these two drugs, chloramphenicol and riconcamides, for them their role is that they bind to also 50S ribosomal subunit, but specifically they inhibit peptidyltransferase. And once peptidyltransferase is inhibited, there is no formation of the peptide bond. And this is also the drugs we can see that are inhibiting translocation. The riconcamides and the chloramphenicol inhibiting peptidyltransferase responsible for formation of peptide bond. Lastly is puromycin. And puromycin, for it causes premature termination. Of polypeptide chain. We have seen for termination to occur, we need a stop codon, but when you take puromycin acts as a stop codon, or it acts as a stop codon whereby it terminates the polypeptide chain before its maturity, leading to formation of a wrong protein. And these are the inhibitors we can talk about. These ones inhibiting the 30S, these ones affecting the 50S ribosomal subunit. Thank you so much for listening. Please always remember to subscribe. Always share your comments, and always hit the like button that we may continue to grow. Thank you so much.